Now let's discuss about DNA mutations after studying about this gene expression. So there are many ways of making errors in DNA, which can be clinically important in some cases. So what causes DNA mutation? First, induced mutation can come from chemical carcinogens or chemical. So for example, ethidium bromide is a chemical typically used in the molecular biology laboratory. And this is DNA intercalating agent for showing up in electrophoresis. Or radiation such as UV light can cause mutation. And there are small scale mutation and large scale mutations. Let's think about small scale mutation first. First of all, when we replicate DNA, it's done by DNA polymerase enzyme. And not all the enzymes are error free. It's just a non-zero error rate. For example, DNA PAR has one error per 10 to 100 million base. So while working hard, pretty precise, but it can still causes mutations. And this single nucleotide to replace for another is called a point mutation. So we know from the degeneracy of this genetic code that sometimes this mutation of one single nucleotide for another can lead to the same amino acid. So in that case, you can't see this mutation happen as a phenotype. So that we call as a silent mutation. But sometimes this change of a single nucleotide for another makes a different amino acid. And that's kind of a problem, and we call it as missense mutation. While there could be even a more deleterious event, which means when right in the middle of this translation, and actually this mutation can cause some of code into a stop codon. Then the translation is making, like producing a polypeptide chain for a protein. It stops in the middle. So it makes a truncated polypeptide chain, which will be a non-functional protein. So, and can truncate. In this case, it's even severe. We call it a nonsense mutation. So there are diseases involved by this single gene point mutations, which are discussed on next slide. And also there are large scale mutations as well. So some genes can duplicate multiple times in the chromosomal genome. So that we call amplification. So it leads to multiple copies of all chromosomal region. For example, this amplification may lead to higher gene expression, which can be a problem. For example, if there's a growth factor or cell division can be accelerated by this amplification, which can cause to tumor or cancer formation. Or on the other hand, some genes or region can be deleted. Here's a good example. So single chromosome with a mutation. So deletion happens, this area, this region is gone, deleted. Then this is a big size, a large scale mutation. Or you can see the same area of this can be duplicated. So this can lead to amplification mutation. And even another case is inversion. So this chromosomal region DNA is just reversed. So inversion, which will lead to a problematic when we translate into making a protein later on. Also, there's insertional mutation. So let's say chromosomal 4, chromosome number 20, and some part of this region can be cross-linked, can be gone into inserting here like this. That's insertional mutation. And there's also translocational mutation. So chromosomal number 4 and 20, if they were like exchange this part each other, so this can be translocated. That can cause a big problem. So this is a large-scale mutation as this, if they happen to create a fusion protein, and that fusion protein leads to a problem for such as keep generating cellular activity or kinase activity, which can be a problem. So that is called juxtaposition mutation. And this potentially form functionally distinct fusion protein. And in fact, in medical world, there is an example, a famous example of chronic myelogenous leukemia. Leukemia is a cancer of the white blood cell. And this is coming from BCR-ABL gene, and this is coming from different chromosome number 9 and 21. Due to this juxtaposition or translocation, they happen to be a fusion protein which will become causing this leukemia. And sometimes this loss of one layer can, in an organism, by deletion or recombinant event, so the expression will be different, and we call it as a loss of heterozygosity. Now, thinking about, depending on the location of the mutation, that effect can be limited to the one generation of oneself, or it may lead to the progeny. So that depends where the mutation happens. So if it's a zoomatic mutation, such as one of our body part, it will affect only my cell. However, if this mutation happens into germline, such as a sperm or eggs, and that part mutation can be inherited to our descendants, that can become a big problem. So that's why these germline cells are more like protected. So let's thinking about point mutation a little more deep. So from the RNA base, which is composed of 4 nucleotides, GCAU, and we read it as a codon triplet. So codon number 1, 2, 3, which will lead to amino acid, a single amino acid, alanine, threonine, glutamate. So this process is called translation. And let's see from DNA until this protein level, these different kind of point mutation can lead. So for example, DNA levels is TTC, and we know mRNA corresponds is complementary AAG, and that can be read by, and then the protein level, this amino acid is lysine. What if this last letter of C is mutated into thymine, and thymine goes to change mRNA level AAA, and if you look at the table, in fact, these two mRNA codons code for the same amino acid. So you don't know what's happening. This is silent mutation. On the other hand, there could be a missense mutation. Let's say this central T, middle T is changed into C or G, then the mRNA level is complementary AGG, ACG, which correspond to create arginine and threonine, which is different amino acid than previous. So this changes an amino acid, and that point mutation is called missense. Another interesting one is at the DNA level, let's say this first T, thymine is replaced with adenine. Then what happens to the mRNA is UAG, which happen to be stop codon. Then once this mutation happens, the translation stops over there and not translate the rest part. So it will create a protein which is truncated, very likely to be non-functional. So that is called nonsense mutation. So in the clinical aspects, these DNA mutations can imply for a number of genetic disease. So first of all, these are charts of with the color showing that the amino acids biochemical properties as non-polar, polar, acidic, and basic. And triplet nucleotide repeat, some diseases are based on this, and deletional mutation, such as cystic fibrosis, deletional mutation can cause this cystic fibrosis, a genetic disease. And another, this green arrows makes this alanine into threonine. And you see the color change, which makes this property change, and this causes prostate cancer. And this aspartic acid mutation, single point mutation causes, make it valine. So this causes a colorectal cancer and sickle cell disease. So there are genetic disease due to this missense mutation. And there's also a nonsense mutation, such as glutamine becomes UAG, which is happen to be stop codon, and that causes a very devastating beta thalassemia disease. And this one arginine into stop codon, tryptophan into stop codon, that is nonsense mutation, which causes a very important genetic disease. So next time we will discuss about gene cloning. Thank you for your attention.